about a lot of scientific papers is having this kind of broad to specific to broad again structure. And today, we've only been talking about the introduction. And one of the functions of an introduction, besides getting the reader prepared to understand the research or experiment that was done, is to go from the broad, big question down to why we would ask this specific question, do this specific experiment. So they do a lot of that in this introduction. So who can get me started on what's the biggest, broadest question that they address in the introduction? How do they open or set this up? Yeah.
Do humans influence diversification?
Yeah. And from there, they have to get the reader to understanding why these finches on the Galapagos is a good study system to answer this question. But there's a more specific version of this that they have to get us to as well. So how do they make this a little more precise? How do they make it a little more focused of a question? Yeah.
They break it up into two ways humans can influence diversification. The first one being humans can cause species to go extinct. And then the second one would be humans cause like divergent species to fuse back together.
Yeah, incipient species, diverging populations. They keep using the term species even though they're not actually two species of Geospiza fortis yet, it's just Geospiza fortis. But they talk about these incipient species fusing. Are any people to think of another possible way humans could influence diversification that they didn't consider? How is this going to affect an overall trend of diversification?
Like you much diversification for all that.
Right. You're going to reduce full diversity and prohibit certain lineages from producing daughter lineages if they go extinct. How's the loss of two incipient species fusing into a single fully interbreeding population going to affect? Yeah.
Because we could have had two new species that could have had their own lineages, but now we can't because there's only one species.
Good. Yep. So those are and so these are both in some way, without using a value judgment of positive or negative, they would be negatively affecting diversification or the potential for it, right? Can you think of another way humans could potentially influence diversification?
Selective breeding.
Why do you say that? Explain a little bit.
Because like with dogs, you know, we want like the best traits with dogs, so we'll breed this set, we'll select traits with another set that we want and get what we're trying to shoot for.
Good. So what, if I can build on that a little bit, what Kane's getting at is he sees more diversity among domesticated dogs in terms of their morphology than in wolves, their ancestors. Of course, all domesticated dogs, even though they vary in their phenotypes, are not different species. But you could imagine that, yeah, there's some negative ways. There could be positive ways as well, right? There could be things that humans do inadvertently or advertently that could potentially increase diversification rate. And that's not something to even put up here. But I just want you guys to think about the universe of possibilities. Okay, and then why the finches? Why Geospiza fortis? Well, that's two questions. Why the Galapagos finches for this study? Yeah.
So because they have 14 different variations.
And what does that tell us? Why? So there's 14 different species of lots of things, but why the 14 of the Galapagos?
Because there's different climates that they had to adapt to.
And how long do we think they've been adapting to these different climates and food sources and habitats on the Galapagos? You guys have a sense of that?
Millions of years?
You can say
think at least
a few million years.
Yeah, a few million years. There's kind of an upper limit on it because we know the age of the Galapagos. We have a sense of when the finches arrived. And there's many different lines of evidence for when that happened. But actually, let me change adapted to to change the adapted to diversification. So there's lots of populations that are each adapting to their own food sources and climates and habitats. And that seems to be a recent and ongoing process. So if you want to catch species that are incipient and see if they fuse back together, you have to find incipient species. And the current ongoing adaptive radiation is a good place to look for such a thing. So that's why Galapagos finches. Now, why Geospiza fortis? Anybody who hasn't spoken up yet first? Yeah.
Because prior to them being around, there were two distinctive beak modes, and they were the ones that started making it less and less distinctive.
And why? Let me turn around. Well, what did they think this bimodal distribution of beak sizes, that Emma just pointed out, was important? What was this a signal of, potentially? Yeah.
They could have went through speciation and become two new species.
Had, you
If
mean
humans were
into the
around
future?
them.
Yeah, okay. So potentially indicates incipient species. Having some divergence into two different populations, into two different phenotypes, a large beak version and a small beak version, indicates they use different resources and may in fact involve incipient, may in fact be related to incipient speciation. What else do you need for speciation? We haven't actually done species concepts yet, right? Anyone have a sense from background biology what makes two distinct species? Yeah. Oh, go ahead.
Me?
Yeah.
Okay. They can't breed with each other.
Right. Okay. So there has to be assortative mating. That's another component of speciation. Did they investigate the mating habits of Geospiza fortis at Academy Bay or over Opatero?
Don't think it's,
That's a much larger question and would involve other papers potentially. So here they're just looking at the distribution of beak sizes. I just want you guys to keep that in the back of your mind because we're going to talk about speciation when we talk more about adaptive radiation. So it takes not just divergence of phenotype, but also assortative mating. There are lots of populations of organisms where you have multimodal or bimodal distributions of some phenotype. That doesn't necessarily mean you've got speciation. But we would expect to see some divergence in phenotypes in incipient species. So that's the signal they were looking at. So bimodal distribution of beak size indicates incipient species. All right, and then we asked you guys about the purpose of the first full paragraph on page 1888. Who wants to tell me a little bit about that? Which Emma was starting to, but is there anything you guys want to add for that, for answers to that question? If you think of this, well, what kind of experiment was this? What kind of study was this? Was this mensurative or manipulative? What happened at Academy Bay? What happened at the treatment site? You can think of it that way. I know you all know this. I heard every group talk about this. What happened at Academy Bay? Yeah.
The human population grew.
Yeah. Good. As opposed to the other two locations, which they use as their spatial controls, right? Can someone explain to me what spatial control means to them? What's important about these other two sites? Yeah.
Just to make sure that it wouldn't happen even if humans weren't there.
Good. What's it in that?
There wouldn't be any a lack of diversification or a fluctuation in diversification?
So if Academy Bay started out looking like this, with beak size, right, and then if we look, we could do this as frequency of pinches. What did they predict would happen at Academy Bay with increase in human population? What's, you said it is happening. How might this have changed if their hypothesis that humans were causing fusion of the two populations look? Same kind of graph. You got it. I'm just want you to think of it in terms of a graph.
I guess there'd be 3 instead of, well, there'd be less of a distinctive peak in the middle, like in the middle of the.
Okay, unless we're seeing a valley in the middle maybe,
Yeah.
so, yeah. Something like that, right, where the middle ones are doing better. Or maybe it's uniform, right? Maybe they all do the same, like that, right? So we no longer have a small population of intermediate-sized finches, but instead the intermediate size are well represented, as well represented as the large and small, right? And so at a place where humans didn't increase in population dramatically, how would you expect the distribution to change or not over time? Yeah.
Oh, there may not even be it may not even show the middle part of it sinking.
It could either persist, or it could even, we could lose the intermediates altogether, right? Is that what you're thinking?
Yeah.
Good. So what about this unreplicated circumstance? Why was this a challenge, and what did the authors do to solve this problem? Again, this is something we started to get at, but I didn't ask this question specifically. Yeah.
So like, there's only one Academy Bay where like these finches come in contact with humans, really. So they had to kind of work with that because they couldn't have that's only one area that they could focus on. So they had to kind of do look at it over multiple years instead of looking at multiple sites of the same thing.
Good, yeah, so having that long-term approach is one way to battle the problem of having just a snapshot of a place in time where it's this distribution and then it's that distribution. Do the distributions persist over time? Is it like this for a long time and then it's always like this? Or does it blink back and forth all the time? The long-term nature of the study gives them the signal that it starts like this for many years, and then do we expect to see this for many years as well? Good. All right. Were there things that, and I asked a few of the groups this, were there things that you felt like you needed to understand in order to read this introduction and summarize it that weren't in our questions that we posted on Moodle? What else did you feel like you needed to know?
What adaptive radiation is.
What adaptive radiation is?
Yeah.
Good, yeah. And do you feel like you have a good understanding now? If you were to tell someone, what would you say?
Not really.
I heard it not really. I saw yes nods and I heard it not really. We're actually going to do an assignment on adaptive radiations where you guys have to tell us what it is and give an example of it. But this is the key here. We've got a recent colonization, geologically recent colonization of the Galapagos Islands by a single ancestral finch. And now we have 14 species, and they're all adapted to different habitats, to different diets, to different climates. In fact, I have a picture of that for you. Display. Marston 215. Turn it on. soon as this comes up, you'll see the picture and we'll come back to it. And we'll finish up this adaptive radiation. Any other terms or concepts you felt like you had to understand? I know I've talked about it with a couple groups, and I know Paul touched it with a couple groups as well. Anybody who hasn't? Let's see, we've got a few of these to put up. A couple back there. Paul, you want to hit the back two with a point? Anybody else? Was there any other concept we talked about that you had to understand in order to get this introduction? Yeah.
I had to look up what bimodality meant.
Good, yeah. What this term bimodality? It's another concept that we use in statistics. It's the shape of a distribution. Yeah, that's a critical one to understand this. Yeah, good.
One thing that I had to look up was specification and just kind of like see what it meant in the context of like science.
But in which context did you see that? Help me out a little bit.
The specification was in this on page like 188. Whenever it was talking like before the biomodality.
Let's see. Now, can you help me
Yeah, I'm trying to
understand a
find.
little bit?
Yeah, I'm trying to find where it said it in the text. So I'd seen it while I was reading, so then I looked it up.
You mean speciation, did you?
Oh, yeah.
Speciation. So the process of going from an ancestral species to two daughter species.
Sorry.
Yeah. That's okay. No, that's all good. That's why we have these conversations, right? To get this stuff. Okay, good. So bimodality, adaptive radiation. Here's the adaptive radiation slide. This is a tree. This diagram here is called a tree. It's a branching diagram. And at the tips of all these branches are samples, basically individual finches whose DNA they took a sample of. And then how close they are on this branching diagram relative to these branch points tells you how similar or different their DNA is, basically. That's the short version. So what we see here is 14 species of finches. If you go into older literature, they often say 13. Here's Magnorostris, Phylogenosa, Fortis is the one that we focused on, right? And here it's closely related to those other two. But all of these are apparently descended from a common ancestor that colonized the Galapagos several million years ago. And in the process, over time, we now have finches that are warbler-like and eat mostly insects. We have ones that are parrot-like and eat mostly fruits. Lots of grasping ones that go for grains and insects and fruits. Crushing ones that eat seeds, which is our group of interest up here. So not only do we have many species, but they're adapted to different lifestyles. That's the adaptive radiation. Many species, many different lifestyles. Good. And there's a third thing that we talked about that you had to understand in order to get this paper. Yeah.
Is it the adaptive landscape?
Yeah. How many people kind of had this idea that they meant actually the different habitats and topology of the island like we talked about in lecture last week? Yeah, multiple people. Hopefully, by talking about a selection surface and adaptive landscape in the context of the young adult lizards, you are better prepared to understand the adaptive landscape in this paper. So, when you guys see these figures, we've got our Geospiza fortis, and they might have small, medium, or large beaks. And you've got frequency of finches. At Academy Day, before the population went up, there were lots of small beak finches and lots of large beak finches, and now a whole lot of finches of intermediate size. How could you change this graph to make it into an adaptive landscape? What is an adaptive landscape graphically? What has been on the y-axis of other adaptive landscapes that we've seen? Yeah.
The different notes, like with the horned lizards, we talked about like the different, like the squash mussel
So those were two different phenotypes, and that's why there were two different graphs, because the squamosal and the parietal represented two different x-axes, two different phenotypes that vary, how long one set of hormones is versus the other. But I'm asking, what was on the y-axis? Yeah.
The survival, right?
The survival. And what was the survival kind of a way of measuring? Yeah, good. So you would expect to see a very similar shape to the graph, even if you change what you're actually quantifying on the y-axis. Survival or contribution to the next generation in terms of reproduction. Right? You'd expect the ones that get a lot of food can afford to have a lot of babies and be well represented in the next generation. Seem okay? Other questions? We're about 126. Landscape. Spatial controls. We talked about incipient speciation and incipient species. They talk several times about species, but they're often talking about Geospiza fortis, population with bimodal. Just trying to see if there's anything else I wanted to hit that I didn't get. Paul, anything you'd like to add here?
Yeah, I would.
Yeah.
Everybody get up out of your chair. Tallest people over here, shortest people at the back. Rank yourselves by height.
No, I'm not. Just how I was born with it. You're taller than me. Scan, I'm the tallest of them.
Can I borrow your
Yeah, he's taller than me.
I think I got you, buddy. I think I got you, sorry.
Yeah.
I didn't bring any.
I don't think so. I think you're a little taller than me.
Yes. Quickly, quickly.
All right, you guys got it worked out?
Curve yourself around so you can see everyone.
Yeah.
So you guys push
Oh yeah,
back.
you can curve, that's good. So you can middle part push back. And the tail, you guys can curve up here a little bit.
They asked me over my height,
Give
so
yourself
I'm
some room.
6'7".
So Dr. Jones showed you guys the other day a distribution of humans. I think it was at Stanford or something.
Yeah, you know, I
We're most likely to live.
check that out. I saw one place Stanford, but there's no citation, so I'm not actually sure where it was. There's also one in Berkeley.
So when you look at this, you should notice some things. What do you notice about your distribution of the phenotype height?
Boys are all over there.
Boys are all over there, women are all over here. And there are a couple of pretty tall individuals, and there's a smattering of short individuals. But what is most of you? You're just kind of average. So it's kind of normal.
We'll all be there.
It's kind of normal for populations to be distributed this way. So this is the phenotype. Dr. Judd's been using this term for a while. We have short individuals and we have tall individuals. And this is the frequency of them. A couple of tall individuals, a couple of short individuals, and a bunch of people in the middle. So this is presumably what finches were like at the beginning, except we put beak size in here, large beak, small beak. What the adaptive landscape does is, one way to think of the adaptive landscape, is underneath this, out in the world, there is an adaptive landscape. It's a metaphor that we use, but out in the world, nature basically says, the environment says, small beak finches and large beak finches are favored and intermediate beak finches are not. And what happens over time is this fitness landscape, where 1 means that you live and a 0 means that you're dead, landscape applied to this population will over time turn this population into a population that looks like the landscape in terms of the distribution. So this is something out in nature. What we see in the final distribution of phenotypes is the result of these fitness consequences acting on a particular population over a period of time. Does that make sense? That's why you can get an insight into what the fitness landscape is by looking at the frequency of the phenotype in a population. So
Yeah, good. And you can,
he did the quick change of the y-axis, but I didn't want you to confuse what the process is by which this happens.
that's good, right. And so you can imagine that as this process is happening, there might be many different metrics of fitness, ways of measuring fitness that would do a better job of getting some insight into what this hard to observe thing actually looks like. Survival might be one, number of offspring per individual, other things you could measure.
Normally we think of fitness as being a lifetime reproductive success. How many offspring do you have over the course of your entire lifespan? But a lot of times, especially if you're a long-lived species, you don't have time as a researcher to wait around until something has lived its entire life, and so you do all kinds of other things to measure what we sometimes refer to as surrogates of fitness, things that we think are going to be correlated with fitness in those cases where we can't measure fitness directly. This is the case with turtles. Turtles are amazingly long-lived, and no turtle researcher is going to hang around for 120 years until they know how many offspring that turtle has produced in its lifetime. And so what we do is we might look at 10-year reproductive success or something like that as a measure of fitness. Or if you really don't have time, you're a graduate student, you have to graduate in 5 years, you might only look at a couple of years of reproductive success. So, yeah.
Good.
Don't forget the process in this, how things are morphing over time. That was all.
Good, thank you. All right.
Are you in fitness?
And that's it, right?
Yep, that's it. I'll see you guys on Tuesday morning. And then be prepared to talk about population growth on Wednesday.